Good afternoon, everybody. We are now live on YouTube, also here at our Zoom channel. Today, we are going to have another presentation on the immuno webinar series that will be given by Dr. Fernanda Ferreira Cruz. And Fernanda graduated in medicine with honors in 2011 and got her PhD in physiology in 2013 from the Carlos Chagas Filho Institute of Biophysics of the Federal University of Rio de Janeiro. She was a postdoctoral fellow in regenerative medicine and bioengineering until 2014 at the University of Vermont School of Medicine, and then returned to Brazil and the Institute of Biophysics as a recipient of a Young Talent Scholarship from the Brazilian National Research Council, CNPq. That was followed by a nota 10 postdoctoral scholarship from the state funding agency, FAPERJ. In 2018, she became a tenured assistant professor at the Institute of Biophysics and a National Research Council and Young State Researcher. She is currently the head of the Regenerative Medicine Research Program at the Institute of Biophysics at the Federal University of Rio de Janeiro. She is also a member of the National Network of Advanced Therapies and the treasurer of both the Brazilian Society of Physiology and the Brazilian Association for Cell and Gene Therapy. She has received many awards and accolades throughout her career, such as the L'Oréal UNESCO ABC Award for Women in Science, multiple awards from the American Thoracic Society, and was elected Young Medical Leadership for the Young Medical Leadership Program of the National Academy of Medicine, and as a member of the Young Physician Leaders of the Inter-Academies Partnership for Health. She's a thesis advisor at the Clinical Medicine Graduate Program and at the Physiology Graduate Program. Her commitment to the graduate students is also reflected in her participation in the Commission for Attention to Students' Mental Health. Her research experience encompasses pulmonary physiology, pathophysiology and immunology, regenerative medicine, bioengineering, and intensive care medicine, and produced 85 papers so far. Fernanda, thank you again for accepting our invitation to speak at the immuno webinars, and you have the floor. Thank you very much.
Thank you, Bruno, for the invitation and for this presentation. Good afternoon, you all, and all my students here. We're going to talk about today about the mesenchymal cells and their derivatives. Where are we in respiratory regenerative medicine? So I have no conflicts of interest related to this presentation, and we're going to talk about. Sorry. Okay, so we're going to talk about the bone marrow-derived mononuclear cells versus mesenchymal stromal cells. We're going to talk about the preclinical evidence of both cell therapies, therapy with extracellular vesicles, and also therapy with mitochondria derived from these cells. And we're going to go through some clinical evidence of cell therapy and some potentialization studies. So first, when we talk about cell therapy regarding lung diseases, the main results are regarding the therapy with bone marrow-derived mononuclear cells and also mesenchymal stromal cells. And why have we chosen this two selected group of cells? So first, bone marrow-derived mononuclear cells, they can be extracted from in the same day, and it can be administered in the same day of the harvesting. So since there's no need of culture, they have lower costs related to the administration of the cells. They have lower risk of immunological reaction. Since we use the cells derived from the patient, that is going to be the receptor. And comparing with MSCs, the mononuclear cells, they present higher number of stem cells trapped inside the lungs following intravascular infusion. And when we talk about the mesenchymal stromal cells, they can be extracted from several tissues, such as bone marrow, adipose tissue, lungs, umbilical cords, and several others. And they have been defined by the International Society of Cell Therapy following 3 criteria. They have to be plastic adherent, they have to have positive markers on their surface and negative markers, especially regarding hematopoietic markers. And they have to differentiate into osteoblasts, adipocytes, and chondroblasts in vitro. So in the past, the mesenchymal stromal cells, they were used because of their plasticity. They were able to differentiate into several tissues, but currently, the rationale of using mesenchymal stromal cells are their ability to immunomodulate. They can interact with several target cells, such as cells from the innate immunity, but also with cells of the adaptive immune system. But additionally to that, mesenchymal stromal cells, they have properties because they can induce epithelial repair, they can promote alveolar fluid clearance, they can also induce bacterial clearance, induce neurodegeneration. Also, it can block the tissue fibrosis, inhibit apoptosis. It can also interact with endogenous progenitor cells, activating this regenerative property, and it can also induce neovascularization. So mesenchymal stromal cells, they are a good alternative for regenerative medicine regarding the lungs. I'm going to show you some preclinical data. First of all, we're going to talk about the asthma that is an airflow that is characterized by an airflow limitation accompanied by inflammatory cells around the areas. The prevalence is really high. 3 million people worldwide have asthma, and we also, despite of not talking about the high indices of mortality, we still have a high number of people that die from asthma every year. And the therapeutic management, includes bronchodilators, corticosteroids, but we all know that steroids cannot block the remodeling that occurs in the lung parenchyma, and also it is associated with side effects. 10% of the asthmatics are refractory to conventional therapy. So in this scenario, we still have to investigate some alternative options to treat this disease. So cell therapy has been studied in asthma for several years, including several papers published by our group. And we have proven that mononuclear cells and also mesenchymal stromal cells could act in not severe models of asthma. And in this paper, we wondered if the mononuclear cells and also the mesenchymal stromal cells would have beneficial effects when we treated a severe model of asthma. And also we wondered, because MSCs are found in this heterogeneous pool of cells that are extracted from the bone marrow, that we name it as mononuclear cells. So we wondered if which was the most important cell inside this pool of mononuclear cells that would promote their beneficial effects. Would the effects promoted by the mononuclear cells would be restrictively dependent on the MSC's fraction, or other fraction would be important? So we have developed a model of severe asthma induced by Aspergillus ephei extract aspiration. And in the last day of the disease induction, we have treated these animals and we have harvested 3 days after. So we have extracted the mononuclear fraction through several centrifugation methods, and we have characterized the mononuclear cells we have counted. And in this experiments, what we have done was what we name it as depletion. So we have collected the whole mononuclear fraction from the bone marrow, and we have taken off all of these selective cells, such as the CD45, as leukocytes, hematopoietic progenitor cells, T cells, B cells, monocytes, endothelial cells, and also the MSCs. So we have used biotinylated antibodies, and then after exposing these cells, these cells that were marked with this specific antibodies, we have depleted all of these fractions, and then we have treated the animals with the resulting cells after these depletions, because we wondered if when we remove these specific cells, would the effect would be still there, or would the effect would be still depleted? So this cell would be really important. So in this paper, we have induced asthma. We have treated these animals with PBS, that is our vehicle, and we have treated with fibroblasts, that was our negative cell control. And we have treated these animals with both MSCs and also with the mononuclear fraction. And not the whole mononuclear fraction only, but we have treated these cells, these animals, with the mononuclear fraction after the depletion of the following and what I have just mentioned for you. So what we have observed is that we have quantified the inflammation score around the areas, and we have seen that the inflammation was there in our model, and it was reduced when we treated the animals with both mononuclear cells and also MSCs. But when we have depleted the mononuclear fraction out of the CD45, CD11B, and also the SCA1, that is the marker of the MSCs, we have lost this anti-inflammatory property that we have seen before. And it was also, we could also see the same pattern of results when we quantified the total cell number inside of the bronchoalveolar lavage fluid. So we have a higher number of the cells in the valve, and it was reduced when we treated these animals with both MSCs, mononuclear cells, and some of the depleted fraction. But when we have depleted the mononuclear fraction out of the CD45, CD11B, and also SCA1, we have lost this anti-inflammatory property that we were seeing when we treated these animals with the mononuclear fraction. Not just the total cellularity, but also the differential cellularity was quantified. So we have seen the same pattern in neutrophils, eosinophils, macrophages, and also lymphocytes. And we have quantified some cytokines associated with the Th2 pathway that is associated in asthma, such as the IL-4, the IL-5, and especially in this model, it was a severe model. We have not just the Th2 lymphocytes involved, but we also had the Th17 lymphocytes involved. So we still have a higher amount of IL-17, and it was reduced when we treated the animals with cell therapy, except of those groups that we are still mentioning here. So when we lose the CD11B and also the MSCs from the whole mononuclear cells, we lost all these beneficial effects. So what we could conclude from this first study is that both mononuclear cells and also MSCs, they are able to reduce the peribronchial inflammation and also the inflammatory number of cells inside of the BAL. And the depletion of both MSCs and also the CD11B cells from the mononuclear cells, they can, it interrupts its effects. So when we study actually looking after the MSCs, how do they work? We know that MSCs are very plastic cells. They have several receptors in their surface and also intracellular receptors that can recognize changes in the microenvironment, such as the presence of chemokines, cytokines, and also TLR ligands. And it can also change what they secrete. And we know now that MSCs can interact with other cells through cell-to-cell contact, through the release of soluble mediators, through the release of extracellular vesicles. And currently, we have been seeing that MSCs can interact with other target cells through the organelle transfer, such as mitochondrial transfer. So in this study, I'm going to show you, we have wondered if only the administration of the conditioned media or the extracellular vesicles would have this beneficial effect that we have seen in our study, in our previous study. So we have used the same model here of asthma induced by Aspergillus IFAX pectoropharyngeal administration. And we have treated these animals at the last day of challenge and harvested 3 days after. So in this paper, I have treated my asthmatic animals with PBS, with fibroblasts, and now we have treated with MSCs that were derived from human bone marrow and also with mice MSCs. But here we have not only treated the animals with the cells, but we have treated the animals with the conditioned media derived from the same amount of cells. And also we have treated these animals with the extracellular vesicles derived from the same number of cells. And here we have the second group that is named E. We have treated these cells with EDCI, that is a component that will polymerize around the cells and blocking the secretion of extracellular vesicles, because we were interested about the effect when we remove it, the extracellular vesicles that were released from the cells. So here, just to show you that EDCI really reduces the secretion of extracellular vesicles. And here we can show you that it does not, we can see here in the left panel that the vesicles, they are there when the cells were not exposed to EDCI. And here, when we blocked this secretion, we could find really a low number of extracellular vesicles in their conditioned media without changing their morphology and also their viability. So we have evaluated the lung function, we have evaluated the airway hyperresponsiveness. So we know that asthma is characterized by an increased airways resistance, especially when we expose these animals to an increasing dose of methacholine, that is a bronchoconstriction agent. And what we could see is that animals that were exposed to this protocol, they have a higher resistance compared to animals that were controlled. And when we treated these animals with the cells, the human MSCs, conditioned media and extracellular vesicles, their resistance was reduced to control levels, except those groups that were treated with the EDCI cells. The same behavior was observed when the animals were treated with the mouse MSCs. And we have also quantified the total cell number inside of the bronchoalveolar lavage fluid. And what we see first, that there is inflammation in these animals that was not reduced when we treated them with both PBS and also fibroblasts. And there was a reduction in the inflammatory number of cells inside the airways when the animals were treated with both human and also mice MSCs, and also with their conditioned media and also extracellular vesicles. But when we blocked the secretion of the extracellular vesicles in these animals, the inflammation was still there, especially the EDCI HMSCs, the human MSCs blocking. So here, just to show you that the peribronchial inflammation in this model is really high, and it was reduced when the animals were treated with human and mouse MSCs, their conditioned media, and also with extracellular vesicles. But when we have blocked the secretion of extracellular vesicles with the EDCI, the inflammation was still there. So just to summarize some of these results is that we, both the administration of cells, conditioned media, and extracellular vesicles from human and also murine MSCs could reduce the airway resistance, could reduce the inflammatory BAL cells, and also the peribronchial inflammation. And when we treated these cells with the DCI, what we observed is that there was a reduction in all of the benefits that we have observed before. In a different scenario, still studying the impact of extracellular vesicles administration, I will show you some of our data regarding the emphysema. That is right now the third leading cause of mortality around the world, still associated with the inspiration of cigarette smoke, but also the vape smoke. And we wondered in this paper if MSCs, some of the problems related to MSCs administration is we discussed in cell therapy that autologous MSCs can be used, but also heterologous MSCs can be used. And we wondered if the source of the MSCs that were the donors here for treatment would have an impact in their beneficial effects. So we treated our animals with MSCs that were obtained from healthy versus MSCs that were obtained from emphysema donors. And also not just the cells, but right now we have treated these animals with extracellular vesicles derived from these cells. So this was the work of a postdoc of our lab, Mariana Antunes, and she have developed an emphysema model induced by elastase injection and intratracheal injection. And these animals received the MSCs derived from the bone marrow and also the extracellular vesicles derived from the cells, but not only from healthy MSCs, but from emphysema donors. And what she has done is after the induction of the disease protocol, she has treated these animals, and 1 week after the therapy, the animals were harvested. So first of all, we looked after the differences of the MSCs, comparing healthy versus diseased MSCs, and we have seen some differences regarding the secretion of some important mediators associated to their regenerative properties. And we have not seen difference on the content of proteins collected from these extracellular vesicles, but extracellular vesicles derived from a diseased model, such as emphysema, they have a lower concentration of particles, but they have a higher size of vesicles that were secreted. And here I'm going to show you some of the results that we have done in vivo model of the emphysema. And what we could see is that there is a collagen deposition in lung parenchyma in this emphysema model, and a reduction in the elastic fibers. And it was also reduced, all these alterations were reduced after the cell therapy, both with healthy and also with diseased cells or extracellular vesicles. And here the remodeling was still established. But when we look at after the airway collagen deposition, that there is a feature of the emphysema, we could see that we could only see good results when we administered the cell therapy with healthy cells and also with healthy extracellular vesicles, but we could not see these beneficial effects when we treated these animals with emphysema-derived extracellular vesicles. When we look at after the heart function, the PAT-PAT ratio, and also the right ventricle areas, they are cardiovascular alterations that are associated with emphysema, and only the healthy extracellular vesicles derived MSCs could reduce both of these alterations. When we look at after the lung morphometry and also the lung function, what we could see is that the alterations in the emphysema groups, they were only reduced in animals that were treated with the healthy extracellular vesicles derived from MSCs. Both hyperinflation was reduced, also the mean linear intercept was reduced, and also the number of neutrophils inside the lung parenchyma. And what we could how can we summarize all these results is that MSCs derived from healthy and from emphysema donors, and also EVs derived from healthy donors, they can reduce parenchyma remodeling, but only EVs derived from healthy MSCs could reduce the lung inflammation, hyperinflation, and also cardiac repercussions. Right now, we are studying what's inside, what are the differences, from these sources, from the cells and also from the extracellular vesicles to explain why we could see all of these results. In a different scenario, in an acute inflammatory disease such as sepsis, we have, just to show you some of the data regarding sepsis, we know that sepsis is is a disease associated with a very high mortality rate around the world, and is defined as a life-threatening organ dysfunction caused by a dysregulated host response to infection. And we have wondered in this work if MSCs, the extracellular vesicles derived from MSCs would have beneficial effects in the model of sepsis. And I just want to call your attention here because we have treated these animals with a really high dose of extracellular vesicles. Just to compare here with the cells, we know that in cell therapy scenario, the higher the number of the cells that are administered, the higher are the beneficial effects that we can observe. But if we do give a higher number of cells in cell therapy, it can be associated with thromboembolic events because they can block the little capillary bed inside of the lungs. But extracellular vesicles, since they have such a small size, they are not truly associated with this embolic events, so it can be an advantage comparing with cell therapy use. So in this paper, we have also tested if the source of the MSCs would impact in the extracellular vesicle cell the therapy. So we have tested if the EVs released from the bone marrow, from lungs and adipose tissue would induce a different or better effect in a sepsis model. So for this work, our PhD student Natalia Blanco has induced a sepsis model by CLP that we exposed the secant from these animals. We have done some perforations, and then we have done the ligation in the secant, and it induces multibacterial peritoneal inflammation. We have treated these animals with antibiotics just to mimic what happens in the clinic scenario. And then 24 hours after the disease induction, we have treated these animals with extracellular vesicles derived from MSCs that were obtained from different sources, such as the bone marrow, the adipose tissue, and also the lungs. So the first data I want to show you is our survival rate. That here in the red line, we can see that there is a reduction in the survival rate of the animals that were exposed to the CLP model. So we have a 77% of survival. And the survival rate increased in these animals that received the bone marrow and also the lung-derived extracellular vesicles. But interestingly, when the animals were treated with the adipose tissue-derived extracellular vesicles, the mortality was higher than our disease control. So the administration of the adipose tissue-derived MSCs, EVs, were associated with a reduction in the survival rate, and we wondered why. Before showing some of the explanations that we have looked at after, I just want to show you here some data that we have looked at after the results in several different organs, since sepsis is a multi-organ disease. And we have observed that the diffuse alveolar damage was increased in the animals that were submitted to a sepsis model. And it was reduced when we treated these animals with both sources of EVs. Only the lung-derived EVs were able to reduce the lung inflammation, here the lung neutrophil infiltration, and only the lung EVs were they were able to improve the lung mechanics. Here we can see the lung elastance that reflects the stiffness of the lungs. So it is increased in the sepsis model. It was reduced in the animals that received the lung extracellular vesicles. But interestingly, again, when the animals received the adipose tissue, it was even worse. It was even increased. When looking after the kidney, we could also observe here that there is an increase in the kidney edema. It was reduced mainly when the animals were treated with the bone marrow extracellular vesicles. The IL-18 here was reduced regardless of the source of these extracellular vesicles. And the KIM1, that is the kidney injury molecule associated with kidney injury as its name, it was reduced when the animals were treated with bone marrow and also the lung extracellular vesicles. Here we have looked at after hepatic liver alterations, and we have seen there is an increase in the vacuolization in these animals that was reduced mainly when we treated these animals with bone marrow extracellular vesicles. And also the number of inflammatory cells inside of the sinusoids was reduced mainly when animals were treated with the bone marrow. When we look at after the spleen, the size of the lymphoid follicle was increased in sepsis model and was reduced after the therapy with bone marrow and adipose extracellular vesicles. And just to summarize our in vivo model of this extracellular therapy, we have seen that the therapy with lung extracellular vesicles reduced and improved the outcomes in the lungs, especially. And when we look at after systemic outcomes, we could conclude that the bone marrow derived extracellular vesicles, they have induced a better effects. They have induced better effects in several other organs. Also, that could explain why the survival rate was better in this group. So, why this pattern happened? So we have looked at then at the features of the extracellular vesicles derived from these different sources. So we have looked at the cells. And also at the extracellular vesicles that were secreted by these cells. There was no difference in the sizes of these extracellular vesicles, in their protein content, and also the number of particles that were secreted by the cells, they were the same. So we wanted to look after, is there any difference in the protein content? Which proteins were present in this extracellular vesicle? So we have done proteomic analysis of these cells, and we have segregated them into their groups. So the adipose tissue here, the bone marrow, and also the lungs. And what we could see, I'm just gonna choose here some of the things that we have observed, since we have a list of more than 1,000 proteins that were described inside of these extracellular vesicles. But the adipose tissue-derived extracellular vesicles, they have several ATP pathways that were enriched in these extracellular vesicles, and that are related to oxidative stress and also inflammation. So we have administered the extracellular vesicles in the first phase of sepsis that is characterized by a pro-inflammatory response. So the administration of adipose extracellular vesicles could even increase and get worse all of this scenario. But the extracellular, the message here is that the adipose tissue is not a bad source of extracellular vesicles, but this moment in this model, in this disease development, in this timeline, they did not have proteins that would protect our animals. But in contrast, Some of the bone marrow, they have an increased pathways that are related to bacterial clearance, endothelial cell permeability, to the increase in the tight junctions, reducing all of the edema and also the inflammatory cells recruitment to the lungs. The results here are really complex. I'm not just going to focus on them right now, but more details can be observed in the paper later. And I have told you that currently we believe that MSCs induce beneficial effects through their contact with target cells, such as immunological cells, also structural cells, through the cell-to-cell contact, through the secretion of extracellular vesicles, and also through the secretion of soluble mediators. But right now, we know that the transfer of mitochondria to target cells is really, really, really important for their beneficial effects. So, and why, how do they do that? So MSCs are known to transfer mitochondria to other cell types through nanotubules, junctions, through TNTs, through gap junctions, through the secretion of mitochondria, through extracellular vesicles, through cell fusion. So, and it has been already described to several cells inside of the lungs and also in other systems, such as the cardiovascular system, neurological system, ophthalmological system, and other cell types. And why is this important? Is because the transfer of mitochondria, it increases the ATP production. It can reduce the redox homeostasis. It can induce the redox homeostasis and also recover all the metabolic properties of all of these cells. So in this paper that we have published last year by Luisa, that is a postdoc from our lab, she has tested the therapy with the mitochondria isolated from bone marrow derived MSCs when systemically administered in a sepsis model. She wondered if it would, mitochondria that would be injected would be internalized by structural cells from the lungs, and it could restore all of the lung physiology. So first, Luisa wanted to prove that exogenous mitochondria could be internalized by both endothelial and epithelial cells. And here she has marked the mitochondria with mitotracker green. And here is our negative control. And here we can see some small dots that will represent that endothelial cells in this slide could internalize the exogenous mitochondria. And here in a higher magnification, we can see that the mitochondria that is stained in green, they are really internalized by the cells. Here is our orthogonal projection that's showing that both endothelial and epithelial cells derived from both control and CLP animals, they can internalize exogenous mitochondria. And after showing that they can internalize mitochondria, what do the mitochondria do inside of the cells? So we have done an oximetry to understand what happens in the basal respiration and also the maximal respiration. First here in the endothelial cells. So in the CLP model, in the sepsis model, the basal respiration was reduced. And when we treated these cells with a mitotherapy, it was restored, both the basal respiration and also the maximal respiration. And also the epithelial cells, the maximal respiration was restored after the mitotherapy. Here, Luisa has shown, she has used the mitosocks that will show us the ROS production from the mitochondria that is increased in the CLP cells, both in the upper panels, the endothelial cells, and in the lower panels, the epithelial cells, and it was recovered after the mitotherapy. Here is the quantification of this, what I just said. So after showing that the cells were able to internalize mitochondria and it would restore their respiration, we wondered if the therapy with mitochondria systemically would restore some of the features that were disrupted in the sepsis scenario. So Luisa has treated these animals with mitochondria derived from mammalian MSCs. She has injected in the jugular vein, and she has evaluated these animals 24 hours after the therapy. And what we could see is that the animals that received the myotherapy, they have shown an improved survival rate when compared to CLP model. Also, the CFU, the number of bacteria collected from the peritoneal lavage fluid, and also the lung elastance, so the lung function was better after the myotherapy. When we look at after the lung histology, what we could observe is that there was a reduction in the collagen fiber content inside of the lung parenchyma, also an improvement in the elastic fiber content, also a reduction in the alveolar polyps, and also a reduction in the neutrophil counts inside of the lungs. We have evaluated some inflammatory mediators such as the IL-1 beta, the KC, and they were reduced after myotherapy. And also the IL-10, that is an anti-inflammatory mediator that we generally quantify in our works here, and it was increased after the myotherapy. Also, the KGF, that is a growth factor associated with the regenerative property of cell therapy, was increased in myotherapy animals, but nothing associated with the ROS. The ROS protection was increased here in the lungs. In the liver, we have seen good results also in this liver vacuolization, in the amount of IL-6. There was an increase in the IL-10, but again, it was not relevant things regarding the protection through the oxidative stress. In the kidneys, what we could see is that there was a protection in lung histology, and also some markers such as the IL-18 here, that was a reduction after myotherapy. And here in the kidneys, we could observe that there was an increase in the NRF2 and in the SOD2 that are associated with the protection through for the oxidative stress. So to conclude all of these parts, we have seen that MSCs can interact with several target cells, such as epithelial cells, endothelial cells. It can restore their permeability of the lungs. It can restore the surfactant function. It can block the inflammatory migration of the cells into the lungs through the secretion of extracellular vesicles, through soluble mediators, and also through the secretion, and also through the transfer, better to say, the transfer of mitochondria to other cells. And some of these isolated items can be enough to induce all of these beneficial effects. So I have shown you some of our preclinical data. I'm just going to go through really quickly through our clinical data. So we have used the mononuclear cell therapy in some patients with a severe asthma. Here is a case report that we have seen. We have used the mononuclear cells derived from these patients, and they were non-responsive to the current therapies, such as the bronchodilators, and also to anti-IgE drugs. So these patients received autologous bone marrow cell transplantation through an intravascular pathway. So the bone marrow cells were aspirated. So the mononuclear fraction was isolated through the phycal gradient, and the cells were administered intravenously, and we wondered if they would get into the lungs and also induce some beneficial effects. Just to summarize some of the data, we have observed that there was an increase in the perfusion of of the lungs in 2 of the 3 patients. Here, we can see here is the quality of life index that is very used in the pneumology clinics. It was increased, especially after 30 days after the treatment, but there was no difference after the whole observation time. And I just want to show you and say to you that the asthma control was achieved with the half of the prior dose of inhaled corticosteroids, and also with the LABA that was used, the drugs that were used 12 months after the cell therapy. And both of them reduced the use of the anti-IgE drugs. A different study that I'm going to show you here is we have now tested some of the MSCs derived from the bone marrow in patients that have emphysema, heterogeneous emphysema. I'm just going to go really quickly here. So patients that have heterogeneous emphysema, some of the treatment is the insertion of this unidirectional valve. And this is the current treatment for these patients. But the insertion of these valves are associated with adverse events such as fibrotic lesion around their insertion, and also inflammatory response around this insertion site. So we wondered if when this insertion would be done, if MSCs could block the inflammatory response here and also could block the whole collagen deposition and also improve the outcomes, the clinical outcomes for these patients. So what we could see, we have 2 groups here. We have tested this in 10 patients. The groups that were submitted to the endobronchial valve insertion without MSCs, with placebo. And here the patients that received MSCs associated. So MSCs proved to be safe in this scenario, not just through the analysis of symptoms, but also through the analysis of some laboratory markers. And regarding the inflammatory process, we have quantified the C-reactive protein in these patients through 90 days after the therapy, and it was reduced in the patients that received cell therapy associated with these valves. Also, we have evaluated the body index, that is a very used predictor of morbidity and also mortality in emphysema patients. And the patients, they have a reduced body index especially. And so the cell therapy were related to a reduction in this morbidity and mortality predictor. And it was associated also with a reduction in the dyspnea index. Also, the cell therapy was associated with quality of life indices 90 days after the cell therapy. And just to compare here what we were seeing in experimental studies, this is a meta-analysis that would show that both MSCs in several preclinical data were associated with a reduction in lung and also systemic inflammation, and also associated with better lung function here. But when we have done the same analysis in several clinical trials that were published with MSCs in COPD patients, in emphysema patients, some of them, the majority of them, show no difference in several important outcomes in these patients. So we like to say that sometimes science does not bring what we were expecting, and that's okay. We have to think of what's happening and give it a step back and try to understand the pattern and what's happening here. So our group have been working with several strategies in order to potentialize the effect of MSCs. So we have done some studies that we conjugate gene therapy preconditioning strategies, and also magnetic targeting, such I'm going to show you in the next few slides. So as I just said to you, MSCs are really plastic cells. They can interact with their, I don't know what the heck. They can interact with the microenvironment, so they can have an anti-inflammatory property when they are exposed to an increased level of such as IL-6 with pro-inflammatory cytokines inside of the microenvironments. And when the levels of these pro-inflammatory cytokines are low, they can have right now a pro-inflammatory phenotype. So it can induce the M1, for instance, they can induce activated T cells. So what we have done first as the preconditioning strategy is to expose MSCs to biological samples. For instance, we have preconditioned MSCs with pharmacological agents, with polyunsaturated fatty acids, and also with hypoxia. And here, just to show you again really quickly, so when we expose these cells to inflammatory samples such as BAL from COPD patients, from emphysema patients, we change the whole secretome of these cells. And we have tested this in an in vivo model as well. And we have also exposed MSCs to the fatty acids, some fatty acids. And we all know that it can change their secretome. Again, I have also tested in an in vivo model. And we have also been using MSCs with magnetic nanoparticles made of iron. And we have intravascular administered MSCs. We have put some magnetic device around the thorax of these animals, so the cells would be targeted to our organ, to the lungs in this case. And just to show you that when these animals were treated with MSCs they have iron nanoparticles, the magnetic field that is generated around the thorax of these animals could increase the cells that are recruited to this organ, and it could be associated with better lung function outcomes, for instance. We have been currently looking after the effects, for instance, of hypoxia and MSCs that are extracted from the bone marrow. So we know that in the bone marrow, the tension of the oxygen is really low when compared to our culture devices here. And we wanted to mimic what happens physiologically. So maybe MSCs would have a better impact, a better transcriptome and also proteomics to induce better beneficial effects. So we have tested this in our work from Cassia, that is our PhD student. She has exposed the MSCs to hypoxia. I do not have the time to go through our results here, but she has studied the proteomics in these both conditions through normoxia and also with hypoxia. Proteins, what is exclusive from both conditions, and just to show you that the hypoxia increases the production of proteins associated with immune-related pathways and also with extracellular matrix-related pathways. I do not have the time. I feel sorry about that. I'm just gonna go through it and say to you that not just the cells, but the extracellular vesicles derived from the cells, they have enriched pathways after being exposed to hypoxia related to immunomodulation and also with extracellular matrix. Just to conclude this talk, since we have been doing several studies associated with the potentialization, we know that when we manipulate the cells, we change it. So we have to keep in mind that regulatory agencies such as the FDA, Anvisa, and also the European Society, they admit that we have to minimally manipulate our cells in order to go for the clinical use. So processing means that we cannot alter relevant biologic characteristics of cells and tissues. So when we are doing all these potentialization studies, we have to keep that in mind. We still have several questions to be answered here. The number of those, which is the best moment, which is the best source, what is inside of these cells, and also with the extracellular vesicles. This is a field that we are really developing in our lab. And I just want to say thank you to all of the components of our lab, our professors, Dr. Patrícia Rocco, Dr. Pedro Leme, our technician, Andrea, and also all of the PhD students, and also the postdocs, and also the undergraduate students that are associated with all of this work. So I just want to thank again the Immuno webinar for the invitation and for being here. It's a pleasure. Thank you. Aí eu posso parar de compartilhar, certo?
Thank you very much, Fernanda. I would like to first open any questions to the students present. Either they can ask directly their questions, or they can put them in the chat.
Can I make a question?
Yeah.
Okay, thank you. I have three actually. I would like to know regarding the sepsis part where you were using extracellular vesicles to treat your sepsis model. I was thinking that, did you use all of the extracellular vesicles from the tissue, or it was isolated from an MSC from that tissue?
Yeah, we have isolated the MSCs from that tissue, and then we have cultured them until their passage 4 to 6, and then collect the extracellular vesicles from the MSCs.
Okay, maybe that wasn't very clear for me, I'm sorry. And the second question is, does these cells, they secrete these vesicles by themselves, or do you have to make a stimuli or something, or they just naturally secrete them in vitro?
They naturally secrete the extracellular vesicles, but we know that when we stress these cells, they can secrete even more. So what we do is that we deplete the culture from fetal bovine serum for, it depends on the protocol, but for instance, for 12 hours or for 24 hours without changing their viability.
Okay, and my last question is, regarding the last part for the translational studies that you were using in human, I was thinking that if you were interested in maybe tracking the cells for a longer period of time to see if they were like populating other organs or tissues, and if they can help in other way, like differentiating and becoming a part of a tissue population themselves, and not just by secreting something. You know what I mean?
Yes. Yeah, thank you for this important question, Henrique, because before doing the clinical studies, we have to do biodistribution studies. So, and also in our preclinical data, we have done the technician marking of the cells, and then looking after with PET scan in the animals. And we also do the biodistribution cells in humans. But in our hands, we can say that MSCs do not stay too long in the body, so it gets removed in the maximum in our hands for 1 week. We cannot track them anymore. And what happens to them is we believe that MSCs, they are attacked by the immunological system of these patients or with animals. They have been phagocytosed. Phagocytosed by other innate immune cells, and so it also triggers all of the magic that happens after. But we're not really concerned about their, for instance, tumorigenic effects, because they have been described for years, more than 5 decades, with no concerns regarding to safety, regarding to tumors in this context, for instance. But right now we are not believing that MSCs act through their differentiation into other cells. We are really believing that MSCs can act mainly through the secretion or through interfering with other cells that are inside of the receptors.
Okay, Fernanda, thank you very much. It was a great talk.
Thank you.
That's really, if anybody else has any more questions.
I think we have another question from a student in the chat from Nayara.
Yes.
Nayara is asking, is MSC administered by intravascular? It's necessary to do something to direct the AVs to the specific location?
Nayara, this is a very good question, because when we think about the cells administration, we know that the cells, they are chemo attracted to the inflammatory site of the disease, but the receptors that do that for the cells are not present at the EVs all of the time. But we believe that extracellular vesicles can be internalized by several cells that are not our target. And some of the studies that are being developed right now, we can target also not just the cells, but the extracellular vesicles, and try to direct them through a magnetic field, for instance. But we know that the extracellular vesicles, they also are internalized mainly by inflammatory cells. And right now, people that work in the field really believe that immunomodulation is really important. So if they are put in the circulation, they will do what they have to do through other cells and not through, directly in structural cells per se.
I have a question. When you were analyzing the different sources of EVs, the lung EVs had some sort of weird behavior, because they did not come together as a group when while the adipose derived and the bone marrow derived, they came together, showing they had these they had this common content to them. But I was wondering if you guys ever thought about making artificial EVs, and is there any hope for that? If you have like hundreds of proteins in there, can you imagine in any situation that you can actually make these artificial EVs that will be ready-made with the different compositions, and whenever you need something that will act in the early phases of the inflammatory response, or in the later phases? How do you actually, if you look into the future, how do you see these going forward?
Yeah, first we have to recognize that MSCs are really heterogeneous. Sometimes we do not have the same results, and we have to work with our protocols in order to increase the reproducibility of the data first. So you have looked after that, and it's really important. And right now we are thinking, trying to understand which is the best source for a moment and for a context of the disease. And we are thinking about inhibiting some of these pathways that are increasing in some type of the extracellular vesicles in order to increase the contents that we believe that is important. So we are trying to bioengineer a cell first, and then collect extracellular vesicles with some things that we believe that are important. Singularly chosen or we can potentialize the components of these extracellular vesicles through some strategies such as the hypoxia conditioning, that it increases a lot several of important pathways that are associated with our beneficial expected effects. So this is a hot spot field and a very complex field as well. Yes. And also not just the proteins, but the microRNAs that are inside of these extracellular vesicles that is a really hot spot. So this is a very complex. I do not have an answer for you right now, which will be the best for everything, because we're still trying to understand what's happening.
Any more questions? Gustavo?
Hi, Fernanda. Super thanks for giving us the opportunity to know better your beautiful work. I'm sorry if I will touch some stupid points. I know that I really have to go and read all your papers. There was a lot of information. But first thing, I'd like to go back a little bit in what Nayara touched, but more now regarding to the mitochondrial administration. So I understood that you inject intravenously. Is that right?
Yeah.
So
Yeah.
how this end up going to the endothelial cells in the lung? And is this the only place? I mean, have you looked to other issues? And also regarding concentration of the mitochondria injected to the specific localization in the lungs?
Very good question, Gustavo. Actually, in our in vivo studies, we have not tracked where the mitochondria went. In our in vivo, we do not do that, but we do believe that mitochondria can be internalized by several other cells, and not just endothelial and epithelial. In our first part of the paper, we just wanted to show that the endothelial and epithelial cells derived from the lungs would be able to internalize them. And in the literature, there are several groups that study that mitochondria can be internalized by innate immune cells, such as monocytes and also macrophages, and it can change everything. It can change their phenotype, their secretion, their inflammatory response. And in a sepsis model, in the first phase, we believe that mitochondria are internalized by them as well, but we do not show in this paper. And I do not have a true answer for you to say that, oh, the endothelial and epithelial, and we have not looked at in this paper, but we believe that it can happen, but also other cell types can also internalize them.
All right, so the image that you show was in vitro internalization. Oh, okay.
Yes.
The
first part, yes.
Right, but you did show experiments with in vivo injection of mitochondria, and then a lot of anti-inflammatory effect. In one of the figure, if I not wrong, you show also decrease in bacterial number circulating, or is it circulating bacteria?
It was in the peritoneal lavage fluid.
Yeah.
After the CLP, when we treated.
Right.
Yes. So
how is this possible? I mean, how does this happen?
Yeah, so there is an interesting paper that has shown that MSCs could transfer mitochondria to macrophages, for instance, and it would increase their phagocytic capability. So we have seen, we do not show in our hands that it happened, but we believe that their data corroborates to our data, and it can change everything. So
right. So if there is a decrease in the number, of available bacteria, do you think that this may also contribute to the effect in the lungs?
Yes.
Or do you believe? Okay, so you believe that also could be kind of a primary target
Yes, because reducing the bacteria load, it can reduce all the systemic inflammatory system activation and also the permeability and everything else that happens after sepsis.
All right, excellent. So if I may ask a couple more questions. One is related to the effect that you show that is sometimes is very comparable, the vesicles, the supernatant, and the cell, the injection. But I think you show us a window that is like a short-term window, if I remember. So do you have any idea on how these three main sources, let's say, of mediators would influence in the long term? Would the cells be better for like long-term effect compared to supernatant, for instance?
Yeah, we believe that MSCs could have a longer effect, but the evidences are really short right now. So we believe that extracellular vesicles and also the conditioned media would allow us to give multiple times to the animals without any immunological risks, for instance, but we believe that MSCs could induce a longer effects, but because the cells would be there. But we know now that MSCs do not stay there too long. So we have several groups that defend one and several that defends the other point of view.
No, excellent. And two quick questions. One is that in the very beginning of your presentation, you showed that the depletion of CD11B cells has an even higher effect or negative effect. In some cases, even exacerbate your inflammation compared to SCA1. So could you comment a little bit on that? And also, finally, you did the proteomic analysis, but could you comment a little bit more on maybe lipid mediators as well?
Yeah.
Thank you.
Yeah, so just to start talking about the CD11B, so when we deplete the mononuclear fraction extracted from the bone marrow of the CD11B, so we believe that when we remove monocytes and dendritic cells derived from the bone marrow, we lose our effects. So we know that in the bone marrow microenvironment, there are MSCs, and we know that MSCs can induce monocytes and also dendritic cells to have anti-inflammatory phenotype. And right now, it was a really great surprise for us because we were thinking our main hypothesis in the beginning of that work is that only the depletion of the MSCs would have a negative impact. But when we have depleted the CD11B, we lost it. And my PI said to me, repeat that, please. And I have repeated, and he said to me, repeat that, please, again. So because we were not believing on what was happening. So right now, our hypothesis is that inside of the bone marrow, they have an anti-inflammatory property. And when we administrate these cells, they can have beneficial effects, not just the stem cell, the adult stem cell, but right now the immunological cells. And you have asked then after the lipid mediators. So we have a conjunct students, a PhD students, that she was trying to understand some of the lipid mediators that were associated with MSCs therapy, but we know that they are important. They have resolving production, they have prostaglandin production, but we have not done like a lipidomics analysis because we do not have the hands and the time and the budget and everything else. But we know they are important.
All right. Okay. Thank you and congratulations. It's wonderful, wonderful work. Thank you very much.
Thank you for the opportunity. I'm really happy to be here.
Bruno is outside. Hi, Gustavo. Hi, Luisa. Hi, everybody. Thank you, Fernanda, very much for your beautiful presentation. With 30 minutes of your presentation, you had a huge body of data being presented already. It was really massive. I was really interested about your strategy using that drug that I don't remember the name right now, that block the release of the yes, this one. And if I'm not wrong, part of in one condition for one of your cells, the block of the EVs are important, but for the bone marrow was not important.
So it was really important for the human MSCs, but the mouse MSCs, we still have some effect. So we believe that mouse, when a murine MSCs was administered into a mouse model, they could act through cell-to-cell content and through some other ways without counting just the extracellular vesicles secretion. But the secretion of extracellular vesicles was really, really, really important in this human MSCs.
I see. Yeah, I thought it, but after that, the main research body was concerning mouse models and EVs. Yeah. So I was wondering, how do you conciliate that? I mean, because I mean, the cells should be much better and more important, more robust model for the models and the marine models. How do you think about that?
Yeah, actually.
And when you're talking about lipids, you know, I don't think about lipids inside of the EV. So I was thinking that they may be the main thing.
Yeah. So right now there are several groups that are enthusiastic of the cell therapy and groups that are enthusiastic for the extracellular vesicle therapies, but we all recognize their advantages and also the disadvantages of both therapies. And we know that there is not a perfect strategy. And now we're trying to understand if we can combine both of them and which moment, which dose, which, you know, that. And regarding the lipids, we're still trying. Yeah. This is a very complex area, and some groups are really specialized in this lipid mediators, but it's not our case from our group.
Yes. And concerned mechanistically, like other people asking here before, and your intravenously injected EVs or mitochondria and how they reach the right cell and are integrated in the cells. But you know, this, I don't know if this is wrong question, but this should happen. I know that you said that this happens in vitro, but this should happen in the blood circulation. You know, the EVs or the mitochondria should get inside of a cell, in the circulation. So then the cell are carried to the right. Have you looked at that? I mean, this is happening.
Yeah, we have not looked at that, but we do believe that they can interact with the circulatory cells, and the circulatory cells will do their job right now too.
I mean, I think this is a question to study, that it's very interesting because we don't think about this happening in circulation usually. Cells usually need to be, you know, adhered to something to do this job, to integrate something.
So there is a paper that shows that MSCs can transfer mitochondria to circulating lymphocytes, for instance, and they have made a movie out of that, and it's so beautiful.
Movie?
A movie, yes.
No, but I know that's a movie, but they are.
Yes. Yes, and it's a beautiful, they have set up everything there, and they have shown that MSCs can transfer through circulating cells, and it's beautiful to see.
I want to see that.
Yeah, I can show you.
Thank you.
But it's also, it was shown before that mast cells, for example, that are in the tissue, they are not isolated from the circulation by the endothelial cell. It's not a perfect barrier, because the mast cells actually can emit projections of their cytoplasm to the circulation and capture their IgEs in the circulation. So I I expect that it can also happen with EVs or mitochondria, and it's probably not something that is exclusive to mast cells. Probably macrophages can also do that. Say any cells that are on the periphery of the vasculature probably can emit these elongations to the lumen of the vessels and capture whatever is circulating there. So well, that is something that's important to study. But well, do we have any more questions? Anybody? If not, I would like to thank again Fernanda for this wonderful talk. It was very exciting. Thanks. I'd like also to thank everybody that was with us today in the audience. Thank you, Luisa, Márcio, Gustavo, and everybody on the São Paulo side of the immuno webinars. And we'll see you guys next week. Yes. We have
next week we don't have seminars because of the SPA.
Yeah, exactly. Thank you, Luisa. Thank you. Quick as always. Thank you very much.
Thank you.